This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's unsupervised learning.
Thanks for listening to the ungated version of the Unsupervised Learning Podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
You know that genetics plays a huge role in our health, and more people are using genetic testing to determine risk for diseases like cancer for themselves and their kids than ever before. So I want to tell you about Orchid. It's the only company that does whole genome testing for embryos, testing before your child is born. If you're doing IVF, this is a clear choice now because now you can reduce risk for thousands of single gene disorders, including heritable forms of autism, pediatric cancers, and birth defects. Check them out at orchidhealth.com.
Hey, everybody. This is Razib with the Unsupervised Learning Podcast, and I'm here to get nerdy about worms. Maybe not the type of worms that some of you are used to. I know some of you do have scientific backgrounds, and when you're hearing me say worms, with my particular interest, you might already suspect that we're going in a different direction that normie on the street would think. I'm here with my friend, Dr. Erich Schwarz. He's an assistant research professor at Cornell University up there in Ithaca. He is freezing right now, and he's very cold. He is in the molecular biology and genetics department. He is a molecular geneticist, and he has been getting into genomics. He's been around for a little while. Been at Cold Spring Harbor, other places, Caltech, and he has a passion for the worm and what it can tell us. Before I let Eric, talk about himself and a little bit more, I do want to say there's a book I read 20 years ago. It was actually 2005. I read it in 2005 by Andrew Brown, British science journalist. He also wrote The Darwin Wars. And, I used to be friendly with Andrew, and he actually commissioned me to write articles for The Guardian. Google it. You'll see it. I actually wrote articles for The Guardian. Some of you might be surprised by that. And he interviewed me for, I think, BBC in 2010. I've actually never listened to that interview. But in any case, Andrew wrote In the Beginning Was the Worm, Finding the Secrets of Life in a Tiny Hermaphrodite. And, for a little while, I did, as an undergrad, I did work in a worm lab in Patrick Phillips' worm lab. And he was switching from elegans to Romanii, a different type of worm. And so I have a little bit of familiarity with these critters. I've looked at them under the light microscope and tried to culture them. I'm not the best I'm a bad bench biologist. I'm just going to be honest about that. So that was not my future. My future was sitting in front of computers and digital stuff. But still, I have some familiarity with this field. I'm still super interested in it. And I think it's really important for regular people to know what some of these model organisms are. Maybe I'll get a Drosophilist on sometime. This is just a thought. But, the worm, C. elegans, is one of the model organisms that biologists use. Model organisms are, there's a huge community. There are conventions all around these model organisms. And they basically try to use these small, usually they're small, they breed fast, they're easy to culture or store or, breed or whatever, raise. And they give you insights, general biological insights, because biology, in theory, is a science where you can get general insights. It's governed by laws, governed by models and theories that apply across the tree of life. This is an attempt, to be like physics, but that's fine because I guess, I think Eric and I would both agree that there's objective reality, and that objective reality has regularities and patterns. And, science is the game of figuring out those regularities and patterns, why they're coming about, and their contingent causal connections. Just sound a little pretentious there, but, it's all for real here. So Eric, I've been talking a little bit. I kind of want to know, I have I have some glimmers of this talking to you, but talk about how you got to where you are now as a worm genomics person, in terms of your training, back at Harvard, and then you went to Caltech, and you've been around for a while. There was no real genomics kind of at the beginning, and now it's everywhere. So shoot, man, tell us.
So I began really in the most goofy possible way. I was a young idealistic kid growing up in the 1970s, which makes me a bit older than you or some of your audience. And I was reading science fiction. And one of the things I got out of science fiction, which I treasure this day, is a sense that the world is not a fixed place. That, the world we're in now at this moment, that we can feel very bored by and we feel it's very mundane and fixed and static. If you came to this place from a different planet, it would be a science fiction world. That's what Neil Stephenson said was the essence of science fiction. It didn't depend on machines or ray guns or flying saucers or women in bikinis, whatever. It was actually the sense that the world was plastic and that humans could alter it through disciplined effort. And that's the feeling I got from all science fiction books I read growing up. And that's really why I decided to become a scientist. I was thinking about various things. When you're a little kid, you get all kinds of things. What I decided pretty early, just from reading books and thinking about things, was that I wanted to live in the world where I tried to make the world better, and I wanted to do it in a potent way. The 1970s were not a great time to grow up if you were a young, precocious American kid looking at American politics. It wasn't very ideal. Things weren't great 50 years ago either. We had Nixon driven out of the White House. We had all kinds of crud. We lost the Vietnam War. It was not inspiring. And my family, much of my family was Irish American, and the assassinations of the Kennedys had left us thinking if you got a good politician, they'd die. So when I was a child, I thought, I want to make the world better. You can't do it through politics. How do you do it? And I'm saying, I want to become a scientist. I want to learn science and be able to use my intellect and have Archimedes' lever and move the world. And I more or less ended up doing that through a variety of positive and negative adventures in terms of trying various things. I ended up going to Harvard as undergraduate. My very first job was protobioinformatics. There was no word for it at the time.
I got a question.
Yeah.
Did you use quotes and did you cite no, I'm just joking. Go on.
No. I mean, I won't drag us too much in the weeds on that, but I can tell you about when I was my very first week at Harvard, they hand you a pamphlet. And the pamphlet, when I was there as an undergraduate in the early 80s, I'm sure hasn't changed much except a few things like URLs, says, if you quote blocks of text without attribution, you are plagiarizing. If you use someone else's words and you don't seriously rethink those words in your own words, you're plagiarizing. They were quite clear about it. And that's all I'm going to say about that.
Yeah, back to the worm. Back to the voyage to the worm.
Back to happier topics. Yes. So I worked for 1 year I took a year off of Harvard and I worked at UCLA in the laboratory of David Eisenberg on what turned into a published, very proto-bioinformatics project on predicting how proteins float in the membranes of cells. I was a very young undergrad working with David Eisenberg, who did most of the work, but I was his assistant. I was a competent enough assistant that I ended up getting a co-authorship on that paper, which ended up doing quite well. It ended up being one of the more heavily cited papers in the journal Molecular Biology, which before journals like Cell was actually considered a high-end journal. It was a very high-end journal in the 60s and 70s, and it was still quite prestigious in the early 80s. So I ended up having a co-authorship on that. At the time, I had no inkling that I'd end up doing a great deal of this. I loved working with Dave. He was a wonderful mentor, an amazingly patient mentor with very precocious me and sometimes obnoxious me. So what that project entailed was simply looking at proteins, looking at the string of amino acids that makes proteins, and trying to predict intelligently what part of protein would sit inside the membrane of a cell rather than inside the cell or outside the cell. And this kind of work was not done a great deal. You could occasionally do it, but people didn't do it a lot. It wasn't something you did every single moment of the day. So I got involved in that and I enjoyed it. And then I went and tried to actually do real science, which meant working at the lab bench with living organisms. And as an undergraduate, I ended up working in the laboratory of Matthew Meselson. Those of your listeners who think more about molecular biology will recognize the name. Meselson did what some people call the most beautiful experiment in molecular biology. He and Frank Stahl showed through direct sedimentation in an ultracentrifuge that DNA truly has two strands. And they're big, intact strands. He was able to show this in ultracentrifuge with a Seisman gradient as a graduate student at Caltech. Many years later, he was a professor at Harvard, and I ended up working in his laboratory. He had moved at that point to Drosophila fruit flies, and I spent a year doing Drosophila genetics in his lab. And that was my first real taste of working with my own hands on genetics of living, breathing organisms. And I found I really enjoyed it once I got past the first month. The first month, that there was a reasonable desire that I show whether I was be good enough to stay in the lab. And it was kind of a hazing for the first month. And I was kind of overwhelmed, but I really wanted to work for the guy. I really wanted to work in that lab. So at the start of the summer, I worked in his lab. I told myself, I'm going to work for 90 days. If I can't stand it after 90 days, I'll stop. And fortunately, after 30 days or so, I felt better. And what I ended up doing was becoming very familiar with fruit flies. So you've mentioned, Razib, model organisms. The reason why model organisms were a huge deal in the 20th century and still matter now, as you've talked about enormously at great length, we are now able to treat humans in some sense as a lab animal. We can sequence literally at will any DNA from any human. We can do population genomics analyses of humans that would have been unimaginable a few decades ago or even a decade ago. We can do all kinds of powerful things with humans now. And this is a huge growing field, and it's very importantly so. One of the reasons we can do this now, we can do direct biology on humans now, is that first of all, we have the tools to actually cheaply get 1 billion base pairs out of a human, which we had to develop. And then the second reason is when you get those base pairs and you map them onto a reference DNA sequence genome, you actually know what you're mapping onto. And the reason you can do that is humans and fruit flies and C. elegans worms and even E. coli bacteria and yeast cells are all one common ancient family. And they have protein coding genes and encode proteins in all of them. And when you look carefully, you can see the marks of ancestry in all of them. You can see relatives of hemoglobins in fruit flies and Drosophila. You can see every major protein in humans, with very few exceptions, has an evolutionary relative in a fruit fly or in a C. elegans. Now you can start to do serious genetic experiments on mammalian cells with things like CRISPR and get somewhere. But
I want to just pause right here. I want to jump in and then I'll let you continue. I want to reiterate what you just said there. Charles Darwin had there was kind of like an assumption, a presupposition of common descent of the tree of life. But this was an assumption, a reasonable assumption, but it was an assumption. And then what happened with the modeling, the unlocking of the structure of DNA is you actually looked at the substrate, at the biochemical basis of inheritance. And when you looked at the biochemical basis of inheritance, everyone's like, damn, this is all the same. It's all DNA.
It's source code, common source code.
And you look at the DNA, it's not common source code, and it has an original kernel. There's a core there. And that core is common to a lot of organisms. And so Darwin's conjecture, because it was a conjecture, was totally validated on the biophysical scale. And so I think that's an amazing fact that we take for granted. But, it was kind of a big deal in the late 20th century when people were like, wow, yeah, our assumptions were totally correct. They went looking for life in the crust and other places, and there were no other alternative types of life. Life is one. So you're talking about CRISPR and stuff. I just want to jump in and reiterate that for that listener.
That's directly relevant, because the whole thing is when you first wanted to understand life at all, I think biologists have always want to know how we humans tick. That's very proper, very natural. If you go back enough decades, you see people like Max Delbruck choosing literally to work on the viruses infecting E. coli bacteria, phage. Why did Delbruck do that? Not because Delbruck was a goof who wanted to pick a weird organism. At that moment in time, in the 1940s, it was literally the most complicated living thing you could hope to try to understand. It was at the bleeding edge of rational understanding. A bacterial virus phage was your absolute bleeding edge of something you might dissect at the bench and understand what's going on. And then when Matthew Messelson and Franklin Stahl did their beautiful experiment showing physically you could show two discrete unbroken strands of DNA in a DNA helix, they went to E. coli. And as Matt later told a man who wrote a beautiful history, Horace Judson, The Eighth Day of Creation, he said, that felt like going to a mouse, because Matt Messelson had been trained in the phage genetics school, and he was used to thinking of bacterial viruses about the level of complexity you wanted to work with. When he and Frank Stahl decided to work on E. coli, they were being kind of brave. They were going to a bigger, complicated thing. And it worked, and it was great. By my time in the early 1980s, as an eager undergraduate, Matt had moved his lab to Drosophila melanogaster, as had many of the original molecular biologists of the original generation of molecular biology, because at that point, the frontier of the limit of what you could understand had moved beyond E. coli to things like brewer's yeast, cerevisiae, which you make beer with or bread with, to fruit flies, Drosophila melanogaster, to Caenorhabditis elegans, the nematode worm I'll tell you about a bit. A few other simple organisms were being used. Mammalian cells were being tried in culture, but fundamentally, in biology, you always push, if you're trying to learn things that are really valuable, You try to go to the limit of what can be done and no further. And it's a matter of judgment and luck what that limit is, but that limit moves with time. So the model organisms for much late 20th century were the limit. And their scientific justification was, if you want to understand how animals grow, how do organs develop, how do we get from a single fertilized undifferentiated egg to a complex human or any kind of complex creature? How do cells move? How do they locomote? How do they duplicate their genomes without breaking things? How do they pump ions in and out? How do they do all these wonderful things? You have to go to the simplest instance of the thing you're trying to understand. And that's what model organisms all are, attempts to pick good organisms in the lab that are simple as manageable, but show you the principle you want to learn.
And
I
so,
ended up doing that.
okay, so I'm sorry I interrupted because simple, manageable principles you want to learn. I would say that I think the different model organisms have different utilities. So, Drosophila melanogaster, I mean, Mendelian genetics and Drosophila melanogaster are basically joined at the eye stalk or something, I don't know what, or like at the, at the antennae.
The very first genetic map was Drosophila by an undergraduate named Sturtevant at Columbia. Yeah. He worked one night in his dorm room and did the first genetic map of what ended up being chromosomes from fly mutations. Yes,
and also this is like going back to grad school and like talking to Mel Green, the late Mel Green. Melanogaster also have these massive chromosomes, right? They have some of these the salivary gland ones, right? And so there's some biophysical reasons that they're useful, right?
The biggest reason, I'm glad you brought that up. I almost forgot. It's been a long time. I'd forgotten it. We can now go to a genome browser on a web page and see a DNA sequence to infinite tail of anything we care about, such as humans. In the early 1980s, if you wanted to look at the genetic material, if you wanted to actually see the genetic content of a living animal, Drosophila has this strange thing where in salivary glands, it's growing larvae, its chromosomes, instead of dividing in cells, duplicate in place. They stay in a single cell, but they essentially overcopy themselves. In a normal eukaryotic nucleated cell, such as a human or ear of corn or a fly or whatever, you have a single nucleus. Inside, you have one pair of chromosomes for each chromosome member, right? In the salivary glands, this process goes nuts. And you end up with a single nucleus of salivary gland with perhaps 1,000 copies of each chromosome. And very importantly, for reasons I'm not sure we know even now, when these divisions happen, the chromosomes stay very well aligned. What this happens at some point, the chromosomal DNA of Drosophila gets large enough to see in the light microscope. And Calvin Bridges was one of the first people in the early 20th century to actually take these salivary glands, gently and carefully squash them on a glass slide, fix them with acetic acid, get a light microscope image, and you end up seeing what looked like these rather thick ropes with stripes of white and dark coloring. Looking through, I can tell you, it basically looks like gray, and you have dark gray and light gray, but they're very regular gray. They're very regular stripes all along the long chromosome, and there's always the same number unless you've mutated the fruit fly. And importantly, when Bridges very carefully looked at this again and again and again, he noticed that for each chromosome, the pattern of light and dark bands was absolutely stereotypical. If you looked at Drosophila melanogaster and you looked at so many chromosomes of a certain type, you'd always see the same white and dark bands. And what Bridges correctly deduced was that this might be genetic material. These might be genes. We now know he was almost correct. Those are gene regions.
Yeah.
But like for large genes, physically large genes that do things like control development are very large and complicated, like the ultrabithorax complex, which helped unlock the mysteries of how you get body organ determination, how you get body development. The ultrabithorax gene complex in Drosophila melanogaster literally occupies an entire pair of bands. And you can see those bands. And Bridges and eventually Ed Lewis later, one of his successors, deduced that there might even be duplications of genes leading to duplicated bands. So this was before DNA sequencing. If you wanted to look at genetic material, that's what you used. I wouldn't want to give the impression that it was all wonderful in the 20th century and then afterwards everyone was just an antiquary. There are several other reasons why you wanted to keep doing this stuff in the late 20th century. And the big one that motivated Matt Musselson and Seymour Benzer, my PhD advisor who did Drosophila, and Martin Chalfie, my PhD advisor who did C. elegans, was to understand how things developed. We don't really have anything resembling a full understanding of that, I would say, in any organism, but we understand far more than we did a few decades ago. And the reason is we started by taking simple animals and doing classical genetics, just like what you might have done in the early 20th century, but this time in very targeted screens for mutants that caused very specifically altered patterns of animal growth, animal development. So we take a normal Drosophila, it starts as an embryo, it matures as an embryo, it hatches out as a little first stage larva. By the time it's reached first stage larva stage, it's this little ellipsoid with very regular bands of spikes on its body. Nusslein Volhart and Erich Weishaupt helped win themselves a Nobel Prize, earn a Nobel Prize, by doing this very extensive mutant screen where they looked for thousands of embryos of Drosophila where you saw these extremely specific defects in how the embryo grew. And they found all sorts of remarkable things. Like they found embryos where half the stripes of the body were missing, or the stripes, instead of having even and odd stripes, you had only odd stripes, or instead of having a head and a tail, you had two heads or two tails. It turned out that if you look carefully, you could find a single gene in a fruit fly. When you broke it, gave you this very specific defect, remarkably strange defect. There's a reverse logic that we still use now to understand humans that we use in understanding animal genetics in general, which is if you want to find a specific piece of the machinery to make something happen, and you mutate the creature, you get a mutation, and you see a very specific break in the animal, then you do reverse logic. You say, well, the gene I broke is making that thing happen normally. So when I break it through mutation, I get this very specific defect because that one particular thing is broken. That simple logic drove a lot of molecular biology last century, and we still use it. And that's how you could take these fruit flies and these worms and do these classical genetic screens and getting hundreds of things you could say rationally, defined steps, integrated single steps on the way to building whole animals. This was a huge part of the motivation for the late 20th century obsession with fruit flies and worms.
Yeah. Let's go back a little bit now. Not even late, like Brenner, Sidney Brenner, the late Sidney Brenner. He's a character. I actually was at a conference at Berkeley in 2008. You know who Brenner's like? You know who Brenner sounds like in his old age? James Watson. And I'm gonna explain why, because this mofo is shocking, like some of the stuff he said. So we're at a conference, but I don't know, it was about biology, and I think Svante Pabo was there. He was presenting some stuff. Maybe that was a different one, but Brenner was there. I mean, I was there because of Brenner, because he's I want to see, mine eyes have seen the power and the glory type. I want to see in the flesh. I sat up front, and so Brenner's there, and some woman was talking about global warming, and she was a person of some size, not actually exceedingly great size in the modern American context, but there was some size to this individual. And after she was done, Brenner started talking, and he was like, and you know what? She's part of the problem. Look at how large she is. And I was like, what? She should get on a bicycle, and she would use less carbon. And we're just like, oh my god. Is this happening? So I don't know. I got to tell that story. I know it's not flattering, but you know what? Scientists are human, and they have their foibles. And I think as far as horrible, cancelable things go, this is just a rude, senile old man at this point. I don't know if senile is the right word, but he's a rude old man. So this is Sidney Brenner, great, genius, born in South Africa from a Jewish family. I think most of his career was in England. Is that correct?
Yes, Cambridge.
Yeah.
He eventually Brenner became peripatetic and had academic bases across the world. But I think a great deal of the work the world remembers him for was at the Laboratory of Molecular Biology in Cambridge.
Yeah, and he was involved in first of all creating this particular elegans lineage, which was what was from trash bin in England, in Cambridge, in that area. The ones that we use, it's so a lot of these model organisms, they come from one little group. And this is before population genomics, and looking at variation was a big deal. they wanted to understand the genetic basis of some isogenic line, right? And so it's like from some like trash bin in England, I think, is what most of those elegans are from. And they got down to like brute force, like just like the hard work of mapping this organism's cells, working on the development. I feel like the worm, and like I'll let you take, because you're a worm guy. I feel like the worm is like massively important in developmental biology, just like the Drosophila, melanogaster is in evolutionary genetics. But talk about that perception.
So I think everything you said is very true, and I'll carry on with it. So I've talked about model organisms. I've talked about the fruit fly. Most people have some sense what a fruit fly is because everyone's had a fly buzz in their face. And fruit flies are like big house flies, except, in my opinion, prettier. I got very used to. I learned to love the beauty of fruit flies in my undergrad year. My first month or two, I had no idea what I was looking at because the organ was complicated. After I got used to it, I actually started thinking about fruit flies as beautiful. They have a certain beauty. They have a head and a beautiful hexagonal eye, and a thorax and elegant wings, all this wonderful stuff. C. elegans nematode worms are beautiful to the people who study them, but they're not really what I'd call beautiful when you first look at them. They're essentially, when full grown, a C. elegans nematode worm is exactly 1 millimeter, or slightly over a millimeter. It has an S shape. When you typically grow it, you're growing a petri dish with E. coli bacteria for it to eat, and it will swim through that and eat, and they'll sort of gently swim through it. And what you'll notice essentially is the extreme symmetry and simplicity of its outer body. You'll have this little thing that's it's got a bit of thinness. It's not thick and stubby. It's got a mouth that pumps away efficiently. It'll occasionally poop, and on the other end. And other than that, except when you have male and female C. elegans mating in the dish, not much happens except they'll eat and grow.
Well, the vagina anatomy is a big deal in C. elegans.
So the anatomy of the C. elegans vagina is indeed a matter of huge importance, and people ask, well, other than pure reasons why, because C. elegans because it's small enough, and Sidney Brenner organized a ferociously dedicated team of people. You can literally is the very first organism in the history of the world where Sulston and some other people were able to start with a single fertilized egg and sit and work with a diffraction microscope for days and watch cell after cell divide and end up with, when the worm hatched out of its egg, a completely accurate account of where every single cell in the body had come from. Sulston published this work in 1983. It's essentially why he got the Nobel Prize. He deserved it, because for the first time in the history of the world, here was a living, breathing animal where you could draw a completely accurate map of how it got to be a free-moving, free-living first-stage larva. You could draw the entire lineage from the fertilized egg down to the originally hatched larva. And that was a huge deal. You'd never done that. In fact, until Sulston's people did it, everyone thought Brenner was insane to think you could do it. After that comes what's called the post-embryonic development. Worms like us have childhood and puberty, except in our case, it takes us years. In their case, it literally takes 3 days. They hatch from the egg, and 3 days later, they're completely full grown. They do their post-hatching growth in organs that are relevant for adulthood, and the reproductive tract is one of those organs. The great majority of C. elegans worms you ever work with in a laboratory are not, I used the word female a moment ago to be clearer to people who don't do this all the time. 99.9% of C. elegans are actually self-fertilizing hermaphrodites, which is one reason why Brenner chose them. They're very easy to grow. You can take a single C. elegans, put it on a dish, walk away, and in a week, 1,000 of its offspring went across the plate.
Let me make it really explicit for the listener, because I think it's going to confuse people a little bit. So there are asexual organisms like unicellular, like bacteria, that just divide into two. And so obviously when he's saying self-fertilizing hermaphrodites, what Erich here is getting across is, yes, like those asexual organisms, they don't need other individuals there, but what they're doing is they're having sex with themselves.
Exactly.
And so the process of fertilization with the eggs, that's happening, but it's happening in a single organism that has both elements there. And so there's recombination and other, all the standard genetic processes are happening. It's just it's sampling from the same genome. It's like, I don't know, maybe like twins that are opposite sex that are somehow almost genetically identical. I don't know what the analogy would be, but I think I got across like that nuance there because I don't want people to be confused.
Correct. They're not like bacteria where you simply put the bacterium alone and it divides in a asexual way. Not at all. You're correct. What happens is that the C. elegans hermaphrodite is mostly female, but it has extra sperm early on as it's going through puberty. You can call it puberty. That's when its reproductive tract grows. That's when its vulva grows. And that's when you get a brief period where it makes its own sperm, and then it makes its own eggs, and then it fertilizes itself. The development of the vulva was of great concern because it turns out that if you want to actually study a phenomenon, it's easier if you mutate, it doesn't kill the animal. A lot of the mutations you want to study, even Drosophila, are somewhat challenging to study if there's development because a lot of these developmental mutations in Drosophila are lethal. If you got an embryo in Drosophila with 2 heads, that thing's going to die. Okay, a lot of these things are beautiful mutants, but they're a little bit in some sense difficult to study because they're lethal.
Well,
in C. elegans, you get all these beautiful mutations that are post-embryonic. The animal lived fine. You end up with a weird shaped, post-embryonic organ like a vulva.
Yeah, so the developmental biologist, evolutionary biologist Armand Leroy, who wrote Mutants in 2005, and he's been on this podcast. I got to get him on again. He's great. But Armand, he wrote about in mutants like teratology. Teratology is like this whole field of these mutants, and there are human like fetuses. There are fetuses that have to have all these weird things. Obviously, they're stillborn, but occasionally people have preserved them because they thought they were monsters or whatever. So, what Erich is getting across is like, in terms of like contingency and modularity, it's like some organisms seem to break apart a little. Their viability is knocked down a bit more. I think like plants are even further along into modularity in terms of they can be like really weird, like Little Shop of Horrors weird. But that's a different topic. Go back to C. elegans and your little like weird mutant worms.
So in the C. elegans mutant worms that a number of people study, including someone I ended up working for at Caltech later on, Paul Sternberg, you can actually do immune screen where you go and screen the worm for profound alterations of the body that are not lethal because they're dispensable for growth and survival to adulthood. The reproductive tract, the vulva and the genital organs of the hermaphrodite, which is the primary sex in C. elegans, is highly mutable. You can get dozens of mutations that alter its shape. What turned out to be a wonderful coincidence is that both in Drosophila melanogaster, where people like Gerald Rubin were studying development of the fly eye, and in C. elegans, where people like Paul Sternberg and Robert Horvitz were studying development of the post-embryonic vulval development system, a lot of the things they mutated when they finally began to be able to clone DNA and sequence DNA from this newly developing model organism, C. elegans, one by one, as they got the DNA sequences and they looked at them, they found these things were evolutionary relatives of genes directly implicated earlier in that decade in human cancer. So in the early 80s, one of the things you were able to do in a dish with a simple model was take cancer cells or precancerous cells and do all kinds of clever tricks to try to pull out the genes that were driving them towards being cancerous. And people in the early 1980s who drove that work include laboratories of Robert Weinberg and Wigler and others. And of course the work by Bishop and Varmus at UC San Francisco. And what they discovered was that human cells could become cancerous because of mutations in a remarkably small number of genes. A human gene, this is very similar to C. elegans for some reason, by the way. We humans and C. elegans both have about 20,000 genes encoding 20,000 different proteins, with variations produced by complications we can get into if you like splicing isoforms, but basically 20,000 proteins for both C. elegans and a human. What people found in the early 1980s, you could get a cancer cell, you could make a cell go cancerous at times by mutating exactly one gene. And then as they started to work harder on it, they realized you could mutate 2 genes to get a better cancer phenotype. But then in the late 80s, when you start to actually get these mutations in the development of a fly eye, which is dispensable for survival, so you can study it more easily, or in the vulval organs of C. elegans, which are sort of dispensable, so you can get all the mutants you want and start mapping and cloning them, you found that a key element of this was the evolutionary relative of a cancer-reducing gene in humans called RAS, which is implicated in approximately 1/3 of all human tumors. And then another homolog you got was in something called the epidermal growth factor receptor, EGF receptor, which is, again, a steady villain in human cancers. There's a major therapy for trying to treat breast cancer involves essentially using antibodies to block the EGF receptor in breast cancer, for instance, Herceptin. And the reason why this had such a huge impact as these things were being cloned, discovered, which was when I was in young person grad school at that point, these genes were being pulled out, and you could look at the DNA, you could look at the protein, you could stare at it, but no one had any concept how these things interacted. You had one cancer gene, you had another cancer gene, another cancer gene, and people realized they probably cooperate somehow. And people tried very hard to find ways to show this functional linkage. And it was very difficult. A lot of efforts failed. In Drosophila melanogaster and C. elegans, almost at the same time, late 80s, suddenly you had this thing where you already had, from the fact that you could do these classical genetic maps of how different genes alter each other's visible phenotypes, you could make a linkage of the genes based on their biological phenotypes. You could do that using classical genetics from the early 20th century. And then you clone the DNA. And then you find the evolutionary relatives of cancer genes. And in one moment, you have a pathway. And because you find these in two extremely divergent model organisms, fruit flies are not close cousins of C. elegans. And you find evolutionary relatives of the same guys in these highly divergent model organisms, both describing the same chained pathway. It was a very strong and correct inference that the same chain of causality was happening in human cancer, in human cells. This is an enormous triumph for the model organism method. There are many other instances I could pick, and many of them are more contemporary than that, but I think that's a good early one. I think it illustrates the kind of thing people are trying to do.
I want you to talk about a little bit about, and I mentioned this isogenic lines, these inbred lineages, and my training is a lot of it's in population genetics. A lot of my focus is in population genetics. We're interested in variation. There's no variation here. So, talk about, even if there's no variation, what are you getting at? Like, what do geneticists do with these? Basically, they're not clonal, because they don't clone, but you know what I'm saying. There's no variation. Like, what's going on here?
So your point is a terrific point. It's one that people, I think, in Drosophila and C. elegans ignored for a long time. Every one of these lab organisms that people studied consisted essentially of someone going out in a field or behind a trash can, picking one instance of some animal, bringing it back to the lab, expanding its descendants, and passing out the descendants across labs worldwide. People tried very hard to do this so everyone could do the same experiments, have the same material to work on. So the enormous variation of any living species, humans, why do you and I not look like twins? Because there's been genetic diversity in our lineage. Some point, some alleles flipped, and that's good for us. That's good for us humans. Others would all look like robot clones. When you want to work on a model organism in a laboratory, when you're trying to study how some basic biological process works, in the initial case where you're just trying to understand, like, what makes a gene talk to another gene, or how do you get an organ to grow, or why do you get a cancer? In some sense, all that wonderful genetic diversity is background noise that you don't want. If what you want to do is take a normal living organism and break its genes with classical genetics, and then use reverse logic to see what the broken genes are good for, so you can start to understand the pathway you broke, which is what a lot of these things came down to doing. All that genetic variation that's wonderful in human species, interesting in all the other living species on Earth, that genetic variation is unhelpful. It's essentially uncontrolled variation you don't understand very well. And this is why for many years, I think a lot of people in all these model organisms were quite indifferent to population genetics and real world variation. They considered it an obnoxious irritation. It was not helpful. If you're going to do classic experiments, you break things, you don't want a bunch of background static.
Yeah. I think we need to remember before big data, before big computing, that, throwing a bunch of variation and data dredging, which is what a lot of people do right now. And, I think people are getting better about this, but, there's a whole field in genomics, a whole phase in genomics when people were just like generating sequence data and getting information out of it. Well, back in the 1980s, 1970s, we didn't know how many genes were in the human genome, if it was ever fully mappable. People were focusing on, dozens, maybe max hundreds of genes. I actually asked Jerry Coyne once, and I couldn't figure it out, but some historian, some Drosophilist out there, my friend Justin Blumenstiel, I should have him on. I know he listens to the podcast sometimes. He's a Drosophilist. Like in the 1980s, like how many Drosophila genes did we know? Probably like on the order of 100.
What year would that have been? What year was he talking about? Do you remember?
Oh, Jerry Coyne?
Yeah, because I
was thinking like around the early 80s, because I saw some papers and
That's not a crazy number. So I actually, it just so happens a few years later when I was at undergraduate starting to work on flies, there was this fantastic book called The Red Book. It was a compilation of Drosophila mutations by Lindsley and Grell. It was published in the late 60s, and they were still having students like me look at it in the early 80s. It probably listed a few hundred genes, of which perhaps 100 were getting serious sustained study. So that sounds about right.
Yeah. So, I mean, isn't that amazing? Like, because now we know there's 19,000 genes in the human genome. We're looking at variations within families. And so we've come so far. But, you're still in the worm world, and the worm world is now moving into genomics.
Not only did it move, actually, I think, Razib, the reason that inspired you to interview me right now, we are on the 25th anniversary of the very first full genome of an animal being published in December 1998, and that thing was by elegans, ceylanicum.
Yeah.
Going to be done. It didn't go in as an afterthought. Ceylanicum led the way, quite sincerely.
Yeah. Why do the Drosophila people go later? Just because like they had, I don't know, they weren't as technologically oriented. What do you think? I'm just curious. I always wonder that.
Let me try to pick my words with care, because there's some stuff. So there's stuff I should not discuss in public, and there's stuff that I can discuss in public. And what I can say in public was the initial efforts were not successful. And that's probably about it. And then after the initial efforts had failed, it turned out that Craig Venter wanted to compete with the Human Genome Project, and he wanted to push what was a very controversial project, which was to do completely shotgun genome sequencing assembly. A little bit of explanation for some of your listeners. You could almost do this, but I'll do it. When people first started to attempt the task in DNA sequence of whole animals, they did a very methodical approach. They would get a bunch of clones of DNA in the lab. They would find the appropriate ordering of these clones on the known chromosomes of the animal or organism they wanted to study. Then they would sequence the clones one by one, having ordered them physically along the chromosome. That was the ordered sequencing approach that was used for human in the first project by the public project. It was used for ceylanicum, and it was an effective approach in a lot of ways. It had a problem. It was very hard to scale up. Craig Venter had the courage to have people attack the problem differently. They essentially said is, if you have good enough computing power, you don't need to order in your clones. Just sequence your clones randomly and use a computer to assemble them. This ended up being vindicated, but at the time I remember being very controversial. So what happened in the late 1990s was that the Drosophila project had not worked out. And Drosophila people were watching ceylanicum pull way ahead of them and very understandably frustrated. And they actually made friends with Craig Venter, and Venter promised them that if they worked with him, he would do the genome, he would publish it and be freely available. And he kept his promise, by the way. He actually was very honorable about that. But what happened essentially was that the Drosophila project was having problems and got this wonderful back headwind from a man who was, I think probably somewhat still remains, a very controversial and polarizing figure, Craig Venter. Venter is basically why Drosophila was far behind and then caught up quickly. So there were two different routes. There was the route that ceylanicum took, the respectable, slow, successful academic route. And ultimately what saved Drosophila and got it up to speed was the Craig Venter just shotgun everything route.
And that shotgun everything route also, got the human genome sequence way ahead of schedule. I remember in the early 1990s, this was going to be like a generational project,
There was yes, people were thinking that way. And then when they, I think it's fair to say Craig Venter is deeply disliked by a fair number of the academic biological community, but I do not think it's fair to go without saying probably the fact that he provided them with real competition sped things up. I do believe that's likely to be true. I remember that too. It was going to take forever, and then suddenly it wasn't going to take forever because they didn't want to be scooped by Craig Venter. So they just didn't let themselves get scooped by Craig Venter.
Yeah, competition drives people. They got some skin in the game now because they got someone else out there. This is, I don't want to get like ideological, but it's like, I always tell people like, this is why you don't want to deal with the government a lot of the time. Cause like no other option. When you go to the DMV, there's no other option but the DMV. And if they treat you like crap, you got to take it, For
what it's worth, I think that what has made American molecular biology work quite well for a long time is that we've had such a jumble, such a mixture. So we've had very strong public sector investment. We've had the National Institute of Health. We've had a lot of the stuff. I mean, a few weeks ago, I'm not sure we're going to get to it, but I these days I do nothing but genomics. And like one of the most big jobs I had this year was just upload a bunch of genome data onto a public genome database run by the federal government, which is incredibly useful. So the public sector is very important to biology. Public sector funding has been crucial. But I believe the heterogeneity of having many different sources of funding and many different sources of people doing science is also a source of vitality for American biology. I don't think I agree with you. Just having the government be the one source is not good. I'm equally glad that private sector is not the only source either. I think having them jumble together is actually
competition, diversity, kind of like a synthesis, complementation. That's why we have academia, we have the private sector, we have the government. It's good enough for government. No, okay. Go on. What were you saying? Something
after Craig Venner added his element to the picture, one of the things that has to be said about the Public Genome Project was he did his stuff and moved on to other things. The public sector people kept going for another 3 years and produced a much more finished human genome. And then in the most recent remarkable effort in the last year or two, they've actually used extremely new and radical DNA sequence technologies to produce the very first completely gap-free human DNA sequence.
Telomere to telomere, telomere to T to T.
What makes this extremely striking is that to do this, we had to develop when I say we, I mean other people besides me, had developed technologies where you could literally take a single DNA molecule and read off 1 million nucleotide letters in a row at will, low but noticeable frequency. And they also developed technology where you could take 20,000 nucleotides in a row and read them off with high accuracy. Those technologies harnessed together are what we can actually now say for at least one human cell line, you can actually go down a complete DNA sequence of the human with no gaps whatsoever, because some of those regions of human DNA are extremely difficult to navigate. They're extremely repetitive, extremely large. So the public sector, I may have sounded like I was being a little dismissive earlier, they kept going. And they eventually drove this thing completely to as close to perfection as we can do. So there's definitely something to be said for both sides.
Yeah, and so I've talked to you a little bit about, actually a fair amount about your research. So what's going on with genomics now in the current, by the way, what's the size of the elegans genome? It's about like 100 megabases or something like that?
Many years, the correct official size. And by the way, C. elegans was the first genome that was supposed to be gap-free telomere long before human. Okay? And that was officially completed in about 2005 at 100.3 million base pairs, 100.3 megabases. I can honestly say that I was part of a team that helped change that. In 2019, we published a paper titled Recompleting the C. elegans Genome. And we titled that for a reason. We used the technology of the 1980s had been done to get the original genome. We used more modern long-read sequence technologies that become available. And what we found is the original project I was recruited for was supposed to be a completely boring, perfect reproduction of the original genome with higher fidelity. We want to take a single absolute clonal strain because the wild-type strain C. elegans in labs, you're saying there's no diversity. After thousands of labs work for years, you get diversity you don't want. So the idea was to redo the original reference strain absolutely clonally and then sequence again and get a perfect snapshot. That was what I was told, and I believed it. I think we all expected that. The team I ended up joining and actually ended up helping run, what we found was that we redid C. elegans, we got back all 100 megabases, and to our great consternation, we got an extra 2 megabases we didn't want, didn't need, didn't know what to do with, but we're actually hiding. So the actual genome of C. elegans and the official genome are not the same. The official genome was 100.3 megabases. The one we published in 2019 had an extra 2 megabases, and we spent quite a bit of time and effort dealing with that. There is now an unofficial version that my colleagues are helping finish now. It's up to 106. We are hoping very much this will be the last. And it is telomere to telomere using the newer technologies with no gaps whatsoever. And so basically the answer, Razib, is that when you use the newer technology, you see things that people in the 80s couldn't see. You see an extra 6% of DNA, even on something that's supposed to be incomplete.
Mm-hmm.
That's the C. elegans genome. Now, all the other genomes have basically been done not quite as well.
Well, yeah, no, but I got, you know what? Like, I gotta say here, okay, so it's about 106 million, right? That's spaces. We got 3 billion for humans. So in the human, about 2/3 of the genome is intergenic. It's not, transcribed. And then a third is transcribed, but like only about around 1% is part of the exome, where it's like translated from transcribed to translated into proteins. The rest is intronic and all that. Okay, so that's the human genome. There's a lot of repetitive elements, like quote unquote junk, all this stuff. That's why that's why we got a big genome that doesn't do much. Okay, although we don't want to get into it, but there could be like regulatory issues and in terms of how the chromatin is structured. Like there's stuff that there's structure and variation in there that's probably useful that we don't consider. Okay, let's set that aside. Yeah, there's a little genome with elegans, right? Talk a little bit. Like, give me a general sense of its structure. How much of it is genomic? I mean, is exonic, all that stuff.
Much less repetitive DNA in humans. So humans have roughly 50% repetitive DNA as scored by various things. In C. elegans, let's just go back to the official genome because we're not finished redoing the revision. Let's work with the official genome size. Roughly 1/6 of the official genome is repetitive DNA. Almost exactly the same amount of DNA as in humans makes protein. So you have much higher amount of DNA in the genome that's making protein simply because there's a lot less random background non-coding junk in there. You've got almost the same. Remember, I was talking about 20,000 protein coding genes. That's literally true. And some of those genes are remarkably similar to human genes. There is a worm ortholog of the monster human gene Titin. There's a worm Titin. And it's a big gene, several thousand amino acids. So in terms of the actual protein coding content, the worm basically has quite a lot of what the human has with 1/30th less random background DNA that's embedded in. You have a fair number of similar small RNA non-coding things. So I don't know if you ever go into this with your audience, but like I talk about proteins because a lot of the work of the cell is done with proteins, but you also have things like microRNAs. They're very important, tiny regulatory switch molecules that are not protein themselves and control how the proteins get made or not made. Worms and C. elegans, worms and humans share many of those microRNAs. If you look, there's a worm relative and a human relative of a great many different microRNA control genes. And then finally, you have the basic, working class things like the non-coding RNAs that make the structure of the ribosome. The bulk of a protein coding machine called ribosome is non-coding RNA. Worms and humans both have that. So basically, a worm genome looks a great deal like a human genome with a lot less background junk in it. That's one huge difference I'm almost forgetting. In humans, you have the classic thing you see in the microscope or maybe your biology textbooks called the centromere. It's like when you have these cells, when you have chromosomes dividing cells, you see the center thing gets pulled around and spindles dividing cells, and they're called centromeres. Worms have no centromeres. C. elegans worms have no centromeres. They have what's called holocentric chromosomes, which means that for reasons we still don't fully understand, C. elegans nematode worms, their relatives evolved, so the entire chromosome is sort of diffuse centromere. And when chromosomes divide, everything gets pulled apart at once. There's no anchor, single anchor that gets pulled apart.
Oh, the visualization would be totally different.
It ends up looking quite different. It ends up that has practical implications for biotechnology. If you want to use DNA to artificially transform a worm with a transgene, a C. elegans worm would be much easier to transform genetically with DNA than, say, Drosophila, because in Drosophila, you must get the DNA into a native chromosome for it to work and survive. In Caenorhabditis elegans, if you want to put DNA into a transgenic worm, you get a glass needle, you squirt in the gonad, you walk away and let the worm recover. And if the DNA has gotten inside the gonad, inside the egg, what's going to become the oocyte nucleus, that DNA will probably survive and propagate. It doesn't actually have to be attached to a real chromosome to do that. And this is basically because in C. elegans, any DNA molecule is essentially a small chromosome.
Yeah. And so you guys sequenced early. Congratulations. You have a whole community now. You were in Cold Spring Harbor with like Lincolnstein and those guys, right? Early 2000s?
I was actually part of a collaboration including them. I would visit them. So for many years, I took a detour. I was a grad student, I was a postdoc, and I spent 10 years working as an employee of a genome database called WormBase, which was one of the first web-based organism databases to deal with both genomic DNA and biological DNA. And I was the first employee, and I was hired at Caltech to do that. And I ended up helping my boss hire people and organize things and help build up what turned out to be this successful genome database. And Lincolnstein was one of the key people in that project because he was doing the web programming using what was then bleeding edge computer language called Perl that he helped develop. So he was the programmer for the web and for the whole web interface. And for many years, he worked from Cold Spring Harbor on that. Eventually migrated to the University of Toronto.
Yeah, and by the way, just breaking the it's not really breaking the fourth wall, but this is actually how we know each other, insofar as my roommate from undergrad was in that lab, and you know him, and there was a Facebook chain of just discussion about various topics, and then it turned out that I left UC Davis literally the week before you came to. I saw your name as part of the genetics seminar to give a talk in the room that I would always go to, like, listen to seminars of visiting professors or researchers. I saw a poster up with your name as like one of the last things that I saw on the UC Davis campus as a graduate student.
It is very strange.
It's so poignant.
It's very strange that we actually got to know each other and have known each other for a long time, and I've yet to have a good excuse to go visit you there in Austin or anything.
What I can say is, like, I have not. Yes, like, you just like have led on to the audience, and I've not met you in person, which means I do not have. I do not have empirical confirmation that you are not an artificial intelligence, sir. You could be a generative AI before its time. If that is true, though, I think, like, I'm not long for this world, so I think you are a human being.
It is funny how small the world is. That's actually one of the things, like, in the work that I've done, where I've been doing C. elegans mostly, and then other nematode worms. One of the good things about it is you actually get to know a hell of a lot of people don't do that stuff, which is how I got interested in what you do. The whole human population genomics thing, I've never personally done that work, at least not yet. But because I've been rubbing shoulders with people, not you personally, but like I've been interacting with people in the biological community long enough, you get to hear about it. That's one of the things about the whole organism thing. You work on one organism, you get expert on one organism. If you have any interest in biology at all, you spend a great deal of time talking about things that are not your job. And you get to know a lot of people, and that's been a good part of it.
I do want to say, in terms of rubbing shoulders, of course I haven't rubbed shoulders with you. I've never met you. No, but let's talk. We got 10, 15 minutes. I want to just kind of wind it where you are right now. What you're doing. You are actually. We've been talking about the history, which is fascinating, and some Mendelian, like some genetic nerdery, which I'm interested in it. Look, I broke out my salivary gland chromosome knowledge, right? That's a pretty deep cut. I'm legit. Like, I'm not just like a code jockey here. But you're doing some interesting stuff with like some practical applications. It's kind of like closes the circle of making a difference, what you were talking about, why you went into science. Talk about what you're doing now.
Maybe the most important thing I'm doing at this moment is working as a member on a team that is trying to help devise a working vaccine against the parasitic nematode hookworm. And I got to that by gradual stages. I worked, as I said, at WormBase. And then as I was working at WormBase, I made friends with a colleague named Robin Gasser, who was and is in Melbourne working on parasitic nematodes. And about 13 years ago, Robin very kindly invited me to help work in his group on a parasitic nematode genome that they were trying to sequence and assemble, analyze, and publish. And they needed someone to help pitch in and help make it happen. And I was very honored to be asked. And I joined and I worked on this thing called Humulcus contortus infect sheep. I ended up applying for jobs because my then fiance and I wanted to stay together, and she was getting offers of possible faculty jobs. So I ended up interviewing at two places that might have gone. And one of the places I interviewed, I revived a friendship with an old friend and colleague of mine named Rafi Arun at the University of Massachusetts. He's now at the University of Massachusetts at Worcester. Back then he was in San Diego. And Rafi was trying to understand hookworm. And he was trying to understand it using various biological tools to control hookworm in ways that are more effective than we have now. He had been wanting to work with a person who did genomic analysis, which I had at that point learned to do. And he had nobody who was good at it, who was willing to help him. And he and I had always been friends, and we just sort of hadn't talked much. And I gave a talk about the genomics I was doing on the parasite Humulcus contortus. And Rafi said, gosh, that would be great to do on hookworm. And after a bit of discussion, we decided wherever I ended up, we were going to work together. And then, in fact, I ended up moving to Cornell with my now wife. And he and I collaborated long distance ever since then on this hookworm. And what we ended up doing is first getting a good hookworm genome and publishing that. And once we had a good hookworm genome, we started saying, okay, this is very exciting. It's very prestigious to have a nice hookworm genome in a hot journal. What are we going to do with it? Because we both agreed just sitting there and worshiping the genome was stupid. And what we finally decided is we're going to try to make a vaccine, which was very brave because neither of us had any experience. Rafi pointed out, hey, Eric, I have no experience. You have less than 0 experience. And I said, yes, this is true. We're going to try it anyway, right? And he said, oh, yeah. We ended up getting support to have one person work in Rafi's lab on a pilot project to find one protein that might be part of a vaccine. And that succeeded. And we published that one protein. And then we went ahead and got 5 years support to have a whole team of people, which I'm one member, spent 5 years trying to find many different hookworm proteins. The relevance of the genome that I helped do with Rafi is that genome is our baseline. All those proteins, that genome, as I mentioned, we think a lot about protein coding genes. Having that genome and having its protein inventory in our hands and knowing various things about the proteins being made lets us pick targets. And we do a lot of different criteria to try to find vaccine targets, but a great deal essentially is trying to throw away bad possible non-targets. So after 5 years, we have actually been able to biologically test over 30 proteins. And that project has, there's some stuff I can't talk about not because I'm trying to be a jerk, because this is new and it's still nascent and it's not my place to talk about it. What I can honestly say is I'm very proud to have been in that project. We have tested a lot of stuff. We are hopeful. That I can say. So it hasn't since come really full circle. I've ended up helping work with a team to try to make something that if it all ends up going as we hope, one day there will be a vaccine you can give human beings that if you give it to them, make them much less likely to be infected with hookworm, which currently infects some hundreds of millions of human beings to this day. The exact numbers are hard to get, but the minimum number is 170 million people right now.
Mm-hmm.
Rafi would argue that's a great underestimate. He's possibly correct. I don't want to argue the point. It's a minimum 170 million right now.
I mean,
it could be much more.
They were a big thing in the U.S. before public health too, right? I mean, this isn't like some obscure exotic. It was like a big thing in the South.
1 century ago, the Rockefeller Foundation led one of the very first serious systematic public health efforts to eradicate what at that time was completely infested hookworm throughout the American South. So yes, this is not a remote problem. It's something we got rid of basically by having better sanitation and greater material wealth. But a good deal of the world still suffers from it.
Well, I mean, so you've been in the game for more than a couple of decades. I won't put a specific number on it. Let's just say you are a child of the 21st century, spreading your child of the 20th century, spreading your wings in the 21st century. Enjoying the vistas and the majestic possibilities of what humanity could do. How are you feeling about the worm and genomics and where you guys are going to go? Like, what's the next grand adventure with your great Captain Brenner gone now into the great beyond and whatnot?
I think that the next huge effort is going to try to be to build organisms, build living things. They're starting to do that now. There's a project underway now to make an artificial yeast cell, the artificial Saccharomyces cerevisiae project. And they've been literally making synthetic DNA and building synthetic streamlined chromosomes and making an artificial yeast cell. And I think that so I'm drawing back on something that Richard Feynman, the physicist, once said, what I cannot build, I do not understand. That principle, I think, is very broad, applies to many things. We would like to understand life fully. We would like to be able both to control it and to help alleviate human suffering and improve human well-being, and also because simply understanding things makes people happy. It's both the source of potential power of human species and source of delight to human species. So I think to understand living things, we want to be able to start building living things. And if we were going to try to build an animal from scratch, it's difficult for me to think of a better animal to make an object of synthetic biology than the nematode Caenorhabditis elegans. It's, the team I'm working with right now, we're trying to make as accurate and error-free a genome as possible available. We had a draft 4 years ago. The one we get out now is going to be as perfect as we can make it. So you'll have the substrate for the DNA, and you have this great simplicity of the organism itself. So you can actually start thinking about doing artificial synthetic biology on a living thing larger than a yeast cell. I would say that's something people might do. There are many other things. That's my personal thing. There are 100 intelligent ceylanicum people you could ask. They give 100 different answers. So I'm not trying to say this is the one thing. That'd be very wrong.
Yeah. One thing I will say is I wrote this in 2014, I think, in the GSA blog. I was solicited by a friend to write it. But I've been saying for a decade now, we're going from the read technology being primacy in the genome to the write technology, WRITE. And so obviously, we just get right into it and write humans, but probably we shouldn't. So, you're talking about model organisms and their utility. I think they're going to be incredibly useful in the era when humans start to figure out how to, genetically engineer, how to write the genome, how to create organisms from scratch. You're not going to create a human from scratch, but you definitely could create, probably like prokaryotes first, but then, simple metazoans, simple multicellular organisms like elegans. And I think that's what you're alluding to here in terms of possibility of synthetic biology, CRISPR technology, genetic engineering, and just taking genomics and genetic science from being simply a stream of data input to generative science, so to speak. To make an analogy with, because we're all talking about generative AI and all that stuff right now. Well, I mean, genomics is going to get generative genetics, right?
Yeah.
Exactly.
Exactly.
That's what I'm going to call it. I mean, I know there's many other terms for it already, but generative genetics generating new sequences for a new time and a new age. And so we're gonna be talking about elegans, 100 years from now on the Holocast. My great grandson is probably going to be talking with your relatives, cultural descendants about worms. Maybe, I mean, it could be 2 centuries. Like 150 years of the worm is totally possible.
Let's hope so. Let's
Yeah.
hope so.
All right, man. It's been great talking to you, and I hope you listeners enjoyed. Obviously, I do love genetics. I know I talk about a lot of other things with a lot of other people, and I love everything that I talk about. But I think when you listen to Erich, just listen to his voice, and this is someone who's passionate about what he's doing, and would that we all find these sorts of passions, and that's what I wish for everybody out there. I think I found mine. I think Erich's found his, and I think you can hear it in our voices. And so, just go look for that thing. As long as it's legal, of course. All right, thanks, man.
Okay.
Whole genome sequencing is used for adults and children every day to assess risk for thousands of diseases. Orchid, a genetics company led by scientists from Stanford, is able to do this for IVF embryos. Now, instead of waiting for a diagnosis, parents can assess if their embryos have genetic variants known to cause severe conditions before their child's even born. No other tests can detect these issues so thoroughly or so early. So check them out at orchidhealth.com.
This podcast for kids